This is the second lecture, and once again I'm going to divide this lecture into two parts. I'll do the spinal cord in one portion and then I'll do the tracks in another portion. Now let's begin by looking at the spinal cord. The spinal cord begins at the foramen magnum, at the lower border of the foramen magnum, and extends all the way down to the level of lumbar 1 to lumbar 2. So it's either at the lower border of lumbar 1 or upper border of lumbar 2 in adults, and goes up to lumbar 3 in children. The length is about, average length is about 45 centimeters in males and a little shorter in females. This spinal cord lies in the vertebral canal, and as you can see here in this image, it is suspended in the subarachnoid space. If you remember from the introductory lecture, the dura mater comes down all the way, and at the lower border of S2 vertebra, which is here, so this is S2 vertebra, that's where the dural sheath ends. The arachnoid mater follows this dural sheath, and that also ends at the level of S2. And I told you that the spinal cord ends at the level of L1 or L2 in adults and L3 in children. So this is L3 in children. Now the point where the spinal cord ends, that point is known as the conus medullaris, because you can see that the lower end looks like a cone up here. So that point is known as the conus medullaris. And thereafter, all the nerves which travel, they travel in a sheath, and that sheath looks like a horse's tail. So that sheath is known as cauda equina. Cauda meaning tail, equinus is for horse. The reason you have these nerves even after the spinal cord has ended is because the nerves have to travel all the way down and pass out through their respective intervertebral foramina. So you can see that the sacral nerves, for example, will be really long, and they'll come down and then they'll travel through the sacral intervertebral foramina. And then the last nerve, which is the coccygeal, will pass out through the sacral hiatus. So that's why you have all these nerves present within the lumbar system. So this part of the subarachnoid space, which continues below the conus medullaris, is known as the lumbar cistern, and we talked of this when we did the first lecture. Now the point where this the spinal cord ends, and which is called the conus medullaris, from the conus medullaris a thin filament, which is really very little nervous tissue, and it's covered by pia mater. This part is known as the filum terminale, and that passes out, as you can see. So it's lying right in the center. It passes out and pierces the dura mater and comes out and attaches to the dorsal aspect of the coccyx. Now, we've already done the meninges of the spinal cord in the first lecture, but I'll go over them again. It has the three layers that you see in the brain. So it has an outer layer of dura mater. Inner to that is the arachnoid mater, and then you have the pia mater, which is very closely attached to the spinal cord and dips in between the fissures. The pia mater sends out, if you remember, a ligament on either side, which is known as the denticulate ligament. And that denticulate ligament attaches to the dura mater and helps to anchor the spinal cord. However, the passage of these spinal nerves as they come out through the intervertebral foramina, these really provide the best anchorage of the spinal cord within the vertebral canal. In the last lecture, we also talked about how outside the dura mater, that means this space outside the dura mater, which is known as the extradural or also called the epidural space. It's called the epidural space. That space had fat, and you can see the fat over here, and it had the internal vertebral venous plexus, which you can see here. So this is the vertebral venous plexus. Notice how this vertebral venous plexus is communicating with these spinal nerves. And if you remember, I also told you that this vertebral venous plexus communicates with the body of the vertebra through the basivertebral vein. And that's how cancers spread, because the internal vertebral venous plexus has connections on the outside. Over here, it has connections with various abdominopelvic organs and even thoracic organs. So pelvic, thoracic organs. So any cancer there can pass in this way, come into the vertebral venous plexus, which is valveless, so it allows blood to flow in either direction. From here, it can go into the vertebral body, or through here, it can go backwards into the spinal cord and then travel up into the nervous system. Let's look at the spinal nerves. There are 31 pairs of spinal nerves, of which, if you remember the count, these are 8 in the cervical region, and there is a difference. You have only 7 cervical vertebrae, but you have 8 cervical nerves. You have 12 thoracic, 5 lumbar, 5 sacral, and you have only 1 coccygeal. While you had 4 coccygeal vertebrae, you have only 1 coccygeal nerve. So that's the distribution of the spinal nerves. And all of these nerves, with the exception of cervical nerve 1, all of these nerves, they pass out through the intervertebral foramen, and that's what I showed you in the previous picture. So you can see here again, they pass out through the intervertebral foramen. And these nerves, spinal nerves, contain both sensory and motor fibers. So the way these nerves, the spinal nerve itself is actually a very, very short segment. And just to go over it, if you look, this part here is really called the spinal nerve, just that little portion. That part is the spinal nerve. It is connected to the spinal cord by what are known as 2 roots. And these, it's not a single root, there are actually rootlets present. And let me just go back to the beginning picture just to show you. Now, if you look here, this shows you the picture very, very clearly. So if you look, this area here, just at this point, that is the region of the actual spinal nerve. Then it is connected to the spinal cord by, you can see these different rootlets. The one which is present posteriorly, that is called, those are the dorsal rootlets. And collectively we just say dorsal root, but these are the dorsal rootlets. And these dorsal rootlets, if you look further, they are actually connected to this dorsal root ganglion. So this is the dorsal root ganglion or the posterior root ganglion. Posterior and dorsal are synonymous. Then these rootlets which are present anteriorly, these are the anterior or ventral roots. So we call them anterior or ventral. And it was dorsal or posterior. Now, so this spinal nerve is connected to the spinal cord by dorsal rootlets and ventral rootlets. And then this dorsal root ganglion and the spinal nerve, these two are actually present in the intervertebral foramen. And if you look at this picture, it shows you very, very clearly, this is the region of the intervertebral foramen. And in this intervertebral foramen, you have the dorsal root ganglion and the spinal nerve. So as soon as the spinal nerve exits from the intervertebral foramen, it divides into 2 portions, these 2 portions. These portions are known as rami. So the one on the posterior aspect, which is much thinner, is known as the dorsal ramus. And the one on the anterior aspect is known as the ventral ramus. And the dorsal and the ventral rami go to supply the body. The dorsal goes to the posterior aspect of the body, which is the back area. So it supplies the skin and the muscles on the back, and those muscles are the muscles which are very close to the vertebral column. The ventral ramus is larger because it has a greater area of supply. It supplies the entire anterior aspect of the body wall, the lateral aspect of the body wall, and a part of the posterior aspect of the body wall which is not supplied by the dorsal ramus. So you can see that. So it has a very large area of supply. So do not confuse roots. Roots attach the spinal nerve to the spinal cord. Rami are after the nerve has exited from the spinal cord, it is divided. That's where these rami are present. Another differentiating point is, so let's go back to this picture here. Another differentiating point which you should know is that the dorsal root is a sensory root. That means impulses enter the spinal cord through the dorsal root. So when they enter the spinal cord, we call these afferent fibers. So sensory and afferent are synonymous. And if you look up here, you can see from the skin this fiber going in. So this is the direction of this fiber. It goes in this way. It has its cell body in the dorsal root ganglion, and then it enters the spinal cord. So the dorsal root carries sensory fibers which go into the spinal cord. The ventral root carries motor fibers, so it is motor, and the fibers here come out of the spinal cord and they go to supply muscle. So this is the direction of fibers. So they come out, so it begins here, comes out of the spinal cord, and then goes to supply muscle and in some cases glands. So this is a motor root, and this is also known as efferent fibers. So do not confuse afferent and efferent. Now the spinal nerve, therefore, so you can see that the fibers are entering in both directions. So let me just, now just so that there is no confusion, so let me draw this for you. So if I draw the spinal cord, and let me just make it a little better, larger. The gray matter in the spinal cord, and then here is the central canal. This is the central canal. So here you had the dorsal root. I'm only going to draw one root, and that's the dorsal root ganglion. This here is the ventral root, and you can see here this is the short stump of the spinal nerve. This is the dorsal ramus. This is the ventral ramus, and I'm going to just make the dorsal ramus a little larger so that it's easier for me to show you the fibers. Now afferent or sensory fibers from anywhere in the body, so if it is from the front of the body, they'll travel first in the ventral ramus, come here, have their cell body in the dorsal root ganglion, pass through the dorsal root and come into the spinal cord. So you can see that the direction of fibers is this way. Suppose there were sensory impulses from the back of the body, from the back region close to the vertebral canal. They'll come through the dorsal ramus, pass into the dorsal root, again have their cell bodies there, enter the dorsal root and come into the posterior part of the spinal cord. So they also have their cell bodies in the dorsal root ganglion. Again, the direction is this way. Motor fibers, and let's use a different color. Motor fibers begin in the spinal cord, and suppose this was destined for muscles of the front of the body, or say in the arm, they come out through the ventral root into the spinal nerve, through the ventral ramus, and then they'll go to the arm. So you can see that this is the direction of fiber. So this was ventral root and this was ventral ramus. This was dorsal root and this was dorsal ramus. Now if the motor fibers or efferent fibers were destined for muscles close to the vertebral column, they'll come out through the ventral root, enter the spinal nerve, and then they pass out into the dorsal ramus to go and supply muscles of the very close to the vertebral column, and these are known as part of the muscles of the back. So if you look here, can you see that the dorsal root has only afferent fibers? They're all in blue. The ventral root has only efferent fibers, whereas the rami are mixed. The rami are really, when we say, when we talk of nerves, let's say we talk of a nerve like the axillary nerve, or we talk of the femoral nerve, we are really talking of the ramus in most instances, and most instances is the ventral ramus, because I told you it has a larger area of supply. So when we say nerve, we are really talking about this part of it. So you can see the rami are, they are mixed, so they have both sensory and motor, whereas the roots are either motor or sensory. So do not confuse the two. So that's very important for you to remember that. Now what you also see up here in this picture is that you have something which is known as, which are known as rami communicantes. These rami communicantes connect the sympathetic nervous system, which is the sympathetic trunk here. They connect it to the spinal nerves. Now, if you remember, the sympathetic nervous system, its central nervous component was only between T1 to L2. So the connections can come only from T1 to L2. So from T1 to L2, you have fibers which begin in T1, and there are fibers which go all the way down till L2. So they come out, they travel, and remember the sympathetic, which was part of the autonomic, is a motor system. So obviously you will have only efferent fibers. So these efferent fibers pass out, so they pass out in the ventral root, they travel in the spinal nerve, and then the T1 to L2 spinal nerves are connected to the sympathetic trunk by two prongs. The rest of the spinal nerves are only connected by one prong. So here, these two prongs, one is called the white ramus communicans and the other one is called the gray ramus communicans. Now the white ramus communicans, this nerve which has come out from the central nervous system, from the spinal cord, passes out, gets in through the white ramus communicans and comes into the sympathetic ganglion. And then in the introductory lecture of gross anatomy, we saw how it did a lot of things. Now let's say it has to go back into the spinal nerve. It will synapse here in the ganglion. A fresh postsynaptic neuron will start. It will pass into the gray ramus, and then from here it will go both into the dorsal ramus and both into the ventral ramus to supply the blood vessels and the sweat glands and the erector pylorus muscle. Now, because its exit out of the spinal cord can only be between T1 to L2, you have only white rami communicants, you have only between T1 to L2. But gray rami, because that connection is to all the spinal nerves, all spinal nerves have gray rami communicants. So it's very important to know that. So you can see that there are only about 14 white rami communicants, but gray rami, every spinal nerve has a gray ramus communicants. Now let's look at spinal nerves and vertebral levels. And if you remember, the spinal cord ended at the level of, let's say L1, so it ends at the level of L1, but the vertebral column goes all the way down to the coccyx. And now we divide the spinal cord into segments based on those little rootlets. So once again, let me go back to the first slide. And if you look here in this slide, there is, unlike a vertebral column where each vertebra is separate, the spinal segments are divided arbitrarily by how the rootlets are attached. So you can see one set of rootlets is attached here. So this is one spinal segment. Now this is where the second set of rootlets is attached. So we have the second segment. So that's how we divide the spinal cord into 8 cervical segments, 12 thoracic, and so on. So to go back, here we see that the spinal cord has ended at the level of L2, or let's say L1, since I said L1 in the beginning. Spinal cord has ended at the level of L1 vertebra, but that doesn't mean that the L1 vertebra corresponds to the L1 segment of the spinal cord, because this is the lowest level of the spinal cord, so that means that's a coccygeal segment. So we want to know how the spinal cord stacks up against the vertebral column. And so there is a kind of a formula that we attach, and this is the formula which is given. So for cervical, you add 1. If cervical vertebrae, you add 1. Thoracic, upper thoracic vertebrae, you add 2. Lower thoracic, you add 3. And just to explain how this is done, look here. So let me say that this is the vertebral column. So let's say I have the vertebral column and this is the posterior aspect. These are the spines of the vertebrae. So we said for cervical vertebrae, so let me just say that this is cervical 4, so obviously this one will be cervical 7, okay? So the spinal cord which is present inside, and let me just use a different color. So let's say this is the spinal cord which is present inside. So what I'm trying to say is that as the spinal cord is divided into segments by those rootlets, C7 vertebra doesn't correspond to C7 spinal segment. Instead, you add 1. So if you know that the vertebral level is C7, you add 1. So this will be C8 spinal segment. So in the cervical level, if you know the vertebral level in the cervical region, if you know the vertebral level, so the vertebral level, let's say, was C7, you add 1, so the spinal segment at that level will be C8. So obviously, if the vertebral level was C6, the spinal segment will be C7. So now when we go down, so let's say now we have the thoracic vertebrae. So you come down, this way, and then you have your spinal cord 2. So in the upper thoracic vertebrae, which means up to T6, you add 2. So if this was, so let's say this was T1, so this was the T1 vertebra, the spinal segment which will correspond to this, so at T1, the spinal segment corresponding to it will be T3. Now, quite naturally, you're going to wonder what happened to T1 and T2 spinal segments. Now, you have to remember, this is a very arbitrary approximation that we have. So it isn't as if C7 is exactly against C8. So, probably the upper part is against C8 and T1 and T2 kind of get accommodated over here. So it's just a way for us to determine if there is an injury at any vertebral level, and then we think, okay, probably these spinal segments have been involved. So it's not a very exact kind of formula, but it just helps us a little bit. So that's why the upper thoracic levels you add 2. So at T6, the spinal level, T6 vertebra, the spinal level will be T8. Then the next thoracic levels up to T9, you add 3. So at the level of, from T7 to T9, you add 3. So at the level of T9, you have the 12th thoracic segment. And then I've given you a list of how you go down. So you can see that. So here, coming back here, you can see how at the level of T12, it's the lumbar 5 spinal segment, and at the level of lumbar 1, which is where the spinal cord ends, the sacral and coccygeal segments, which are rather small, are all clustered together at that point. So this is how you're going to determine the spinal levels, and this shows you the same thing. So a possible question you could get is that a person has an automobile accident. It is noticed on an X-ray that the thoracic 9 vertebra is damaged. So based on that, which spinal segment do you think would be injured? So naturally, then you'll use this formula. So if you say T9 is injured, so remember lower thoracic was up to 9, so you would think that the 12th thoracic spinal segment is the one that's likely to be injured. So that's how I would ask you questions on this. Now let's look at the structure of the spinal cord, and when you look at the spinal cord you can see there is a little, when I draw it also, I always draw it in this fashion, if you have noticed how I draw the spinal cord. So you have a fissure present in the on the anterior aspect or ventral aspect, and that's known as the ventral or anterior median fissure. And on the dorsal aspect there's just a little sulcus, so that's known as the dorsal median sulcus. So you can see here that's the dorsal median sulcus, and then a ventral median fissure. Then on either side where these rootlets attach, you have small sulci present. So these are known as ventral, and the one at the back over here, so you'll have a dorsal and a ventral sulcus on the lateral side. So these are just known as dorsal and ventral sulci, and this is where the rootlets attach. So you can see how these rootlets are attaching on the sides, and they're often given the name of the anterolateral sulcus. So you can call it the ventral anterolateral sulcus and the dorsal and dorsolateral sulcus. So not ventral anterolateral, you just call it anterolateral sulcus or ventrolateral sulcus and a dorsolateral sulcus. So that's for the attachment of the nerve rootlets. And here again I would like you to see that how you have the ventral root, this is the dorsal root, this is the dorsal root ganglion, and look at the spinal nerve. The actual spinal nerve is just this much, and then of course it divides into its rami. So this part will divide into the dorsal and the ventral ramus. Now let's look at the internal structure of the spinal cord, and I've mentioned this in the introductory lecture where I told you that the gray matter or the nerve cell bodies in the spinal cord is present in the center, and it is surrounded by the white matter which consists of the axons. That white matter is present on the periphery. Now this gray matter, if you notice, is kind of H-shaped, and it contains the central canal, and that central canal is continuous with the fourth ventricle. Now this gray matter is again further divided. You can see it's got little projections. So on the posterior aspect, this is known as the dorsal horn. That's called the dorsal horn. And on the anterior aspect, this portion is known as the ventral horn, or also known as anterior horn. So cells which are present here are known as anterior or ventral horn cells. Cells present here are known as dorsal horn cells. Now again, all the ventral horn cells they contain predominantly motor fibers, or these give rise to efferent fibers, because if you remember, fibers beginning here are the ones which come out and go this way. So the ventral horn cells are the ones which are motor in nature. The dorsal horn cells are the ones which receive the sensory input. So the sensory fibers or the afferent fibers which enter the spinal cord, they go and synapse on these dorsal horn cells. So remember, the dorsal horn cells receive afferent fibers, and the ventral horn cells give rise to motor fibers. Now there are certain nerves which are present, and they are not present throughout the spinal cord. They are present in the cervical region, and two important nerves are the spinal accessory nerve, which goes and joins the cranial accessory, which is the 11th cranial nerve. So spinal accessory. And we also have the phrenic nerve, which is cervical 3, 4 and 5, cervical 3 to 5. So these two nerves, they are present only in the cervical segments they get their origin from the cervical segments of the spinal cord. So in addition to this ventral horn, in the cervical region you'll see cells which give rise to the spinal accessory and the phrenic nerve. And it's a good idea for you've already done the phrenic nerve when you did the thoracic region, so you can see how it arises from the cervical region of the spinal cord. Now in the sacral segments of the spinal cord, you will find special nerves which are present again in the sacral 2, 3 and 4 segments of the spinal cord. And if you remember, when we were talking about the parasympathetic nervous system, we said it had a cranial part and a sacral part. So again, in the sacral 2, 3 and 4 segments of the spinal cord, you can see up here, you have cells present which give rise to these special nerves which are part of the parasympathetic nervous system. Now, in the thoracic and lumbar regions of the spinal cord, the upper lumbar regions, you have an additional horn present laterally, and that's known as the lateral horn. And that lateral horn belongs to the sympathetic nervous system. So it gives rise to the preganglionic sympathetic fibers. So preganglionic sympathetic fibers, just as those sacral ones gave rise to the preganglionic parasympathetic. So this was preganglionic parasympathetic. So here in this lateral horn between T1 to L2, so between T1 to L2, you have, they give rise to the sympathetic preganglionic fibers. And they also receive actually the visceral input. So all the sensations coming from the viscera, which travel again through the dorsal root, because they are sensory fibers, they come and they'll synapse in some cells here in the lateral horn. So that's the gray matter of the spinal cord. The white matter, which surrounds it on the sides, is has fibers which you can see, which are sensory fibers which come in. So these sensory fibers will travel up towards the brain, and they can be present in any part of the white matter. Plus you'll have motor fibers, which are fibers which come down from the brain. So they travel in this white matter. So they are fibers which coming in a downward fashion, and as they come down, some of these fibers then synapse on the anterior horn cells, and then the anterior horn cells give rise to the final output of motor fibers. So in other words, we'll be doing these tracks a little more in detail, so you'll understand it then. So all of this white matter which you see has fibers or axons. These axons may either be traveling upwards towards the brain, or they may be axons which are coming down from the brain and they are present here. So these which go up are known as ascending fibers or ascending tracks. These which travel down are known as descending fibers or descending tracks. Now this white matter is divided into three areas based on the presence of this gray matter in the center. So you have a part present posteriorly, this posterior region. This posterior part over here is known as the posterior funiculus. It's called the posterior funiculus, or also given the name of dorsal column. This portion which lies on the side is known as the lateral funiculus, and then you have a portion which is present in front, that's known as the ventral funiculus. So you have a dorsal, a lateral, and a ventral funiculus. This area which is right in front, which connects the two sides, this area here is known, this part here is known as the anterior commissure. Let me write that here. It's called the anterior commissure. You can just check the spelling. I think there is one M. So that's the anterior commissure, which connects, you can see, the two parts of the two sides of the spinal cord. And it's important for you to remember this anterior commissure, because when we talk of certain tracks being formed, you'll see how fibers cross in the anterior commissure. Now what you can also see in this spinal cord is that in the region of the neck, which is the cervical region, and in the lumbar region, the spinal cord is dilated, and that's where you have more gray matter. And the reason why you have more gray matter in that part is because you have more cells, and these cells are meant for supply of the upper limbs from the cervical area and the lower limbs from the lumbar area. So that's why you have the column, the spinal cord, dilated in that part. Now this white matter in the dorsal column contains entirely all sensory fibers. So in this dorsal column you have all afferent or sensory fibers, whereas the lateral funiculus and the ventral funiculus, these are mixed. So you have both motor and sensory fibers. So this is mixed and the ventral funiculus is also mixed. Now what happens is that the dorsal columns, these parts here which are called dorsal columns, these are, if you look at the sacral and the lumbar region, this dorsal column it's much smaller because it's carrying sensory fibers only from the lower limb and the lower half of the body. And it forms a tract over here which is known as the fasciculus gracilis, or also known as a gracile fasciculus. So fasciculus is another name for white matter. If you remember, axons, collection of axons was also called white matter. It was called lemniscus. It was called tracts. Fasciculus is another name that you use for collection of axons. So in the lower half of the body, and seen in the lower half of the spinal cord, so from the coccygeal up to the lower thoracic region, you see only the fasciculus gracilis, and the white matter is much smaller there. As you go up, then you begin to get impulses from the upper half of the body. So as you are nearing the upper part of the thoracic and the cervical region, you see that the white matter has increased, and now actually you see the presence of two tracts. One is the fasciculus gracilis, which was coming from below, so that remains up here. And then you have the addition of another tract, and that tract is known as fasciculus cuneatus, or also called cuneate fasciculus. So right here you should remember that fasciculus gracilis contains impulses which come from the lower half of the body. So I'm just going to write lower limb. And fasciculus cuneatus contains impulses coming from the upper part of the body. So I'm going to write upper limb. And then we later see what kind of impulses these are. So here it is only sensory. These dorsal, they are collectively called, the fasciculus cuneatus and fasciculus gracilis are collectively called dorsal columns, and they are only sensory. The lateral funiculus contains both sensory and motor tracts, and the ventral funiculus again contains both sensory and motor tracts. Now here what I also want you to notice is that you can see as these tracts keep going up, they actually have what is known as a somatotopic arrangement. That means there's a definite arrangement on how the body is represented in this portion. So we said that the gracile fasciculus or fasciculus gracilis is for the lower limb. So these are the lower limb fibers and the lower half of the body. So if I was to draw these like this, you can see this is the lower limb and the lower half of the body. But when you go up into the cervical region, you can see now that the upper half of the body is also represented. So you can see that now you have the upper half of the body. So I'm trying to draw a person lying on the side like this. And this is the head region. But the head is not yet represented in the spinal cord. And again, when we do the tracks, you'll remember that. So you can see that this is the lower limb. So just so that that's the foot and the lower half of the body, and now you can see the upper limb. And remember, in the spinal cord, the head part of the body is not represented, so that's why I'm going to draw it in dots. So you can see there's a definite arrangement. So if the middle part was damaged, let's say that this area, there was something wrong with this area, you can see that the lower limb will suffer but the upper limb will be spared. Whereas if it was this area which is damaged, then the upper limb will suffer and the lower limb will be spared. So that's why I want you to know the arrangement, and you must know which fasciculus carries impulses from which region. Now let's look at this reflex, which is known as the myotatic reflex, and this is a reflex which you test in the clinics, and this is to test neurological connections. And what it is really a stretch reflex which involves muscles of the same side. So it has an afferent limb. So as you tap this tendon up here, you can see the tendon is stretched. that stretching goes and stimulates receptors which are present within in the muscle, and those receptors are known as muscle spindles. Now, once that receptor gets stimulated, it sends impulses into the ramus that goes into the spinal nerve. Then it goes into the dorsal root ganglion. Remember, afferent fibers always went through the dorsal root. So you can see the cell body in the dorsal root ganglion, enters through the dorsal root and comes and synapses in the posterior horn. If you remember, I told you the posterior horn cells received sensory impulses. Now, once it synapses in the posterior horn, it stimulates, this plus sign is for stimulation. It stimulates an anterior horn cell which comes out through the ventral root, through the spinal nerve, goes into the ramus and comes and synapses on the muscle. And here this muscle is the quadriceps muscle. So the muscle contracts and the person extends the leg. So this kind of a monosynaptic, because there's only one synapse present here, this kind of a reflex is known as a myotatic reflex, and these are the reflexes you test to see the integrity of the nerves. You test these reflexes in the clinic. So, you know, when a patient comes and you want to see whether, let's say, the femoral nerve is intact or that segment of the spinal cord is intact, then you'll test these reflexes. And I've given you some of these segments. When you test the biceps, for example, you're testing the integrity of cervical 5 and 6 segments. When you're testing the quadriceps, you're testing L2 to L4 segments of the spinal cord. Now this you can see this is a monosynaptic reflex, but what I also want you to see at this point is that while this muscle is contracting, its antagonist on the opposite side, which has an opposite action, that is made to relax, and that is done by the same muscle spindle which sent in afferent fibers and stimulated some anterior horn cells. That same afferent sends a branch to another nerve fiber present within the central nervous system known as an interneuron. That goes and synapses on anterior horn cells. These come out and these go to the antagonistic muscle. But the antagonistic muscle is not stimulated. Instead, it is inhibited. So that's why when you contract one muscle, the opposite or the muscle which performs the opposite action, that muscle is made to relax, because you cannot have both contracting at the same time. You'll have no movement. So for movement to occur, while one muscle contracts, the other muscle is made to relax. So that's another important thing you should be looking at in this image. Now, this is not as simple as it looks. So we have this myotatic reflex. So you can see this is the afferent fiber, comes in, synapses on an anterior horn cell. So if you follow this, synapses on an anterior horn cell, that anterior horn cell goes out to the muscle and the muscle contracts. But it is influenced by a lot of other fibers which come from above, or there may be fibers present locally too. And just to show you, in the introductory lecture, I talked about Renshaw's cells. And if you remember, Renshaw's cells were cells which kind of had a braking effect on these anterior horn cells. So if you look at this anterior horn cell, as it's going to supply its muscle, it kind of sends off a side branch which synapses on this Renshaw's cell. And then this Renshaw's cell in turn synapses on that very same anterior horn cell. This Renshaw's cell has that negative braking action on the anterior horn cell. So in other words, the anterior horn cell, as it's sending impulses here, it kind of sends an impulse to the Renshaw's cell, which in turn controls the anterior horn cell. So it makes sure that the anterior horn cell does not get overactive. In tetanus and rabies and some other conditions, these Renshaw cells are knocked off, because at this synapse, glycine is liberated. So when they are knocked off, no longer is glycine liberated. So the anterior horn cell kind of goes berserk and it keeps firing, which is why this muscle acts or rather contracts, repetitively. So that's one influence that the anterior horn cell is under. Then we have fibers which come from the brain itself and from different parts of the brain. And most of these fibers For example, you have corticospinal fibers, you have fibers which come from an area of the brain known as the reticular formation. I mentioned red nucleus, you have these rubrospinal fibers. So you have all of these other fibers which are present here. All of these kind of exert an influence on the anterior horn cell either directly or through an intermediate neuron, and they control the anterior horn cell. So sometimes what happens is when the influence of these is gone, and most of them again exert a braking action on the anterior horn cell. So when that influence is gone, so suppose these are cut, then the anterior horn cell is sort of comes out of that negative or inhibitory effect, and so again its action becomes very brisk. And so you'll see when we do nerve injuries or neuronal injuries, how different reflexes can become very brisk or they can get very sluggish, and that's the reason is because of this control that we see at this point. Now we are going to look at tracts, and tracts are basically a collection of axons which have a common origin. So they begin at one particular point, they have a common function, they all carry the same impulses, and they all terminate at the same point. And as I mentioned in the earlier part, tracts are divided into sensory, which carry impulses to the brain from the body, and then we have motor, which are impulses which come from the brain and spinal cord to the muscles and glands. So sensory are known as ascending because they go up the central nervous system. Motor are known as descending because they come down the central nervous system and then exit. So these are tracts. And before we actually go into tracts, let's first look at sensations. So we'll be doing sensory tracts first. So let's first look at sensations and see how we test these sensations. So we have sensory pathways, and you're going to be testing these sensory pathways. Now let's look at some types of sensations that we see. The first is conscious sensation. That means this is sensation which you can perceive, which you know what is happening. And any sensation that comes from the external world is known as exteroceptive. So these are exteroceptive. They come from the outside world and they impinge on receptors which are present over your entire body surface, or they impinge on the receptors for your vision and hearing. So they are not present on the body surface, but they're a little bit inside, but they're still counted in the exteroceptive. Then we also have visceral sensations. For example, when you feel the sensation of pain in your stomach or in your bladder, and you can feel that, so it comes up to the conscious level. Now visceral sensations are slightly different, in other words that different from exteroceptive, because exteroceptive you can feel touch, pain and temperature, whereas visceral sensations, these are only perceived as pain, and pain is a different kind. It's it's not if you poke the stomach you're going to feel pain. Pain in the viscera is either if the viscus is stretched, so either stretch, or the viscous blood supply suffers, so ischemia. So these are two ways that pain in any organ is felt. Either the organ is stretched, for example, if your intestines are stretched by too much gas, then you feel pain, which is felt as colic. Or if a portion of the intestine or even your heart, for example, is ischemic, then you feel pain at that point, and that is, again, ischemia is what gives rise to the pain. So those are visceral sensations. Proprioceptive sensations are sensations which arise within the body, and they are concerned with impulses which come from your muscles. Those muscle spindles, for example, they carry proprioceptive impulses. They tell the body what the tension, what the position of the muscle is. From your joints, again telling you the position sense. From your bones, so they give you kind of a sense of the body's position, of movement of joints and muscles and tendons, and these are known as proprioceptive. So when you flex your muscle and your joint is when you sort of flex your joint and the muscle causes that action, so you know that your elbow joint, for example, is flexed or it's extended. So that is, it goes up to the conscious level, so that is known as conscious proprioception. Then we have certain impulses or sensory pathways which do not reach our consciousness, and they go to the cerebellum. We are not even aware that these things are happening. For example, your body's posture is being maintained all the time, and you are not even aware of it, because that goes up to the cerebellum, and it is intended for smooth motor coordination. The tension in your muscles, for example, the muscle stops contracting after a point and then relaxes. So you don't even know when it has stopped and when it's relaxing or how much tension has been achieved. So this kind of impulse is known as unconscious proprioception, and this goes to the cerebellum. And then there are certain visceral reflexes which also fall under unconscious proprioception, such as, for example, your heart rate. It will increase or decrease based on the needs of the body, and like the Herring-Brewer reflex, which is in the lung. And you must have done this in physiology, or you will do it. The Herring-Brewer reflex, which prevents the lungs from over-expanding. So you don't know, it doesn't reach your conscious level, but the body takes care of it. So this again is unconscious proprioception. And you cannot test this unconscious, all of the unconscious proprioception. For example, you cannot test Herring-Brewer reflex and so on, but some of it you can, unconscious proprioception, which we'll see when we do the cerebellum. But most of the time in the clinics, what you're really testing is your conscious proprioception. And this is done in alert patients, and these patients have to be cooperative. Now let's look at some of the sensory testing that we do. The first is touch. And you can do touches of two types. One is known as crude touch, and the other one is called fine touch. So in crude touch, for example, you can just take a wisp of cotton or a Q-tip and then brush, gently graze a person's any part of the body and see if the person can tell you where they have been touched. Then you give a person an object and tell them to, whenever you're doing this kind of testing, you always have the eyes closed. One is you have the eyes closed because you don't want visual input to because they can, if you touch a person somewhere and they have their eyes open, they can tell you where they're being touched. So that's why you have the eyes closed, to do away with the visual input. So the other type of testing that you do is, with their eyes closed, you give them some familiar object and you ask them to tell you the shape and the texture. And when you're testing that is known as stereognosis. So you're trying to determine whether they can tell the shape and texture of a familiar object, and this being able to tell the shape and texture of a familiar object is known as stereognosis. So this is called stereognosis. Then for fine touch, so all of this was under crude, the general grazing of the skin or telling the shape and texture of a familiar object. In fine touch, you do something which is known as two-point discrimination. Ideally, how this is done is that you take maybe a divider and it has its two tips. So you take one of these dividers which is like this, and it has 2 points, and you put this on a person's skin, at the part that you want to test. Usually when the distance between them is about 3 to 4 millimeters apart, up to that level a person can detect that there are two separate points. When it gets closer, then you cannot tell that the 2 points apart. But this is very tedious if you have to do it all over to test. So instead what one does is that you draw on the patient's hands. You draw numbers or something on the patient's hand, and you ask them to identify what was drawn. And the purpose of doing this is it tests the ability to recognize distance and also the direction of movement of the stimulus across the skin. So instead of doing this 2 point this way, we kind of draw on the hand, and the reason being that two-point discrimination can get very, very tedious, so we use this. Testing pain is very, very simple. It's like a pinprick, so you prick the person all over the area you want to test and see if they can feel the pain. Temperature is tested by using hot and cold test tubes. Again, you put it against the skin of the region that you want to test, and you ask the patient whether they can tell whether it's hot or it's cold. Then we do tests for conscious proprioception, and we have either active or passive tests for conscious proprioception. So these are all your active tests for conscious proprioception, and this is passive tests. Again, these are done in alert patients who are cooperative, and of course the eyes are closed at this time. In toe the line, you ask the person in the erect position to stand still, and they walk in a line. They sort of toe the line and without swaying. So they shouldn't sway, and then that tells you that they are okay. Now, someone has had alcohol. What happens is that alcohol increases the effect of an inhibitory neurotransmitter, which is called GABA, gamma aminobutyric acid. And I referred to this earlier when we were doing the first lecture. And it decreases the effect of an excitatory neurotransmitter, which is called glutamate. So it increases the inhibitory effect, and because of this, it causes sluggishness in the brain, which is why someone who's taken alcohol tends to sway when you're doing this toe the line test. The other test which you do is a finger nose test. Here the person's arm is extended, again with the eyes closed. You ask them to bring their finger towards their nose, and if all the sensory pathways are normal, they should be able to do this without a problem. In the heel knee test, you ask the person to lie down, so they are in a recumbent position. You place the heel of the foot on the opposite knee and then drag it down and do it again. And again, it tests conscious proprioception, and done in alert patients with the eyes closed. Now, the passive tests that you do is, one is the joint sense. The person has the eyes closed. You grasp the thumb or the big toe and you move it up or down, and you ask the patient to describe whether the movement was up or down, and then you'll be able to test joint sense. Vibratory sense is tested by placing a tuning fork. You either place it on the shin, which is the front of the leg, the anterior aspect of the leg, or the styloid process of the radius. Or the styloid process of the radius, the bone radius on the lateral aspect of the forearm. That's where the styloid process is. Or you place it on the shin, and it's a tuning fork which has been struck, and then you ask the patient if they can detect the vibrations. So that will tell you. If they can't, then you'll know that something is wrong. So this is how you test the sensory pathways. This next slide is just showing you the tracks, and before I end, I want to just give you how the sensory tracks pass up to the brain. So let's look here, and so we are going to describe sensory tracks. So we are doing sensory tracks. Now, all these sensory tracts, they all travel towards the central nervous system. So they're going towards the central nervous system. They go to the spinal cord. From the spinal cord, they go up to the brain. Most of the time, the ones that go up to the brain, they have 3 sets of neurons. So that means you'll see 3 cell bodies. So 3 sets of neurons. Those that go to the cerebellum, they have 2 neurons. So the cerebellar are 2 neuronal pathway. The others which go to the brain are 3. It's a 3 neuronal pathway, 3 sets of neurons. The ones which go to the brain, so let's say this is the brain and this is the cerebellum. So just to tell you the difference. The ones which go to the brain, they always cross over. And I'll show you what I mean by this. So they always cross over. So these are known as contralateral. They'll go to the contralateral cerebral hemisphere. The ones which go to the cerebellum do not cross, so they remain on the same side. So they go to the same side cerebellar hemisphere, and these are known as ipsilateral. So they remain on the same side. Again, a definite orderly arrangement is seen, and that is what is known as the somatotopic arrangement. And I showed you that in the dorsal columns. And again, I said that these are always modulated by other neurons, so they are influenced by other neurons. So first, let me tell you what is meant by contralateral and ipsilateral. So let's say, let me draw a person up here. So let's say this is a person this way. And let's draw the spinal cord and the brain. So, you know, this is the brain up here. And I'm going to, that's the right side and the left side. And then just, you know, go all the way down. That's the cerebral hemisphere, and that's the spinal cord. Okay, so this is the other parts of the brain. Now, let's say that we have an impulse which is going from the, let's say, a pinprick which goes from the arm. So you can see this is the left side of the body. So from the left arm, this impulse will go and it will cross over. So what will happen is that, as I said earlier, there will be 3 sets of neurons. So let's say that there is one set here. Then this will cross over and go to the other side. Somewhere here maybe a second set, and then from here it will go here. So you can see that the left arm is sending its impulses to the right cerebral hemisphere. And I didn't notice that this is the right side, right cerebral hemisphere, and that is the left cerebral hemisphere. So the left arm is sending it to the right cerebral hemisphere. Same from this side, it will go to the left cerebral hemisphere. So you can see, and I also said there are 3 neurons in it. So this is the first neuron. So there's one synapse here. So this is the second neuron which has started, then synapse here, and this is the third neuron which has started. So there are 3 neuronal pathways when it goes up to the brain. Now let's look at the cerebellum. So let me take another thing so that we don't get confused. So let's look at the cerebellum. So again, let's draw our little man up here. And we draw the brain. So here is the cerebral hemisphere, the right and left, and this is the rest of the brain, and this is the spinal cord. And let's say I'm going to draw the cerebellum up here somewhere like this. So this here is the cerebellum. So in the cerebellar pathway, so this is unconscious proprioception. So let's say it's from a muscle up here that's showing you the tension in the muscle. This pathway comes here and it will synapse here, and then it will remain on the same side and it will go to the cerebellum of the same side. So can you see it has not crossed over, it's on the same side. So this is known as ipsilateral. And in the previous one, in this one, this is called contralateral. So the cerebral control, that means the cerebral control is contralateral. The cerebrum always controls the opposite side of the body, whereas the cerebellar control is ipsilateral. It always controls the same side of the body. Here itself, I want to tell you that in the cerebellum at one point when we are doing these pathways, you will see that there is a set of fibers which goes this way. So it goes in and it actually crosses over at one point and it's traveling on the opposite side. And so you think, oh, but it's not supposed to cross. But what happens is it finally recrosses and comes back and goes to the cerebellum on this side. So you can see that it's still going to the cerebellum of the same side. So it still remains ipsilateral. So that's really important for you to understand this. So let's go back to our tracks up here. So in this image, what I'm trying to show you are all these tracks. So you can see here, this is an ascending fiber. So it's from the skin. So let's say it's a pinprick on the thumb. So the fiber will begin, it will have its cell body here. So you can see this is the first cell body, first neuron, comes in through the dorsal root, and this cell body is in the dorsal root ganglion. So the first order neuron, because there are three neurons, we call them first order, second order, and third order. So the first order neuron, its cell body is in the dorsal root ganglion. So you can see here, it's in the dorsal root ganglion. You must know this. The first order neuron is in the dorsal root ganglion, always. Then it comes in here, and now the second order neuron begins in the posterior horn of the spinal cord. Now this can change in some cases. So the second order neuron need not always begin in the posterior horn of the spinal cord. It may begin higher up. But for now, let's just say that it's begun in the posterior horn or dorsal horn of the spinal cord. Notice how it is crossing over up here, and now it's lying in this area of the white matter where fibers from above and below will join it. So that's how it's going to form a tract. And here again there is a definite arrangement, and when we do this tract, you will see that how the legs are represented more on the lateral side and the arms are represented more on the medial side. So I've just drawn the little figure up here. The head and neck is not represented in the spinal cord, even though I've drawn the head here. So now this tract goes up, so that this tract consists of second order neurons. It's traveling all the way up in the brain, in the spinal cord, So this tract travels all the way up in the spinal cord, enters the brain, where it goes and synapses in a part which is known as the thalamus. And now in the thalamus, the third order neuron begins and goes to a part of the brain which is known as the sensory cortex. So you can see that we will say, if we were to take this example, the cell body of the first order neuron is in the dorsal root ganglion. The cell body of the second order neuron is in the dorsal horn of the spinal cord, and the cell body of the third order neuron is in the thalamus. So this is how you're going to remember the tracks. So whenever we are doing the tracks, you must know, first of all, where it crosses. So you're going to know where it crosses, where the crossing occurs. You must know where the cell bodies begin. So where the cell begins or the cell body begins, that is, or where the cell body lies. So for example, the first order neuron, the cell body lies in the dorsal root ganglion. In the second order neuron, the cell body lies in the dorsal horn, or you can say it begins in the dorsal horn. And so you must know that, where each neuron cell body lies. And then you must know where it crosses, and you must know where it terminates. So where it terminates. So, I've already mentioned where the crossing occurs, whether it occurs in the spinal cord, whether it occurs in the medulla, whether it occurs somewhere else. So these are some of the things that you must know when you're doing these tracks. So I'm going to stop here, and I will do the tracks in detail in the next part of this lecture.